Hello and welcome. Today, we're going to do something a little different. We're going deep, deep inside the microscopic world to uncover the secret strategies, the literal playbook that pathogens use to make us sick. So let's crack it open and see exactly how they pull it off. I mean, really think about that for a second. How can something so tiny, so completely invisible, follow such a precise and honestly brilliant plan to infect and overwhelm a host that is trillions of times its size? Well, that's the mystery we're going to unravel today. All right, so before any game can really begin, you've got to know the rules, right? So first up, we need to define our key terms and really understand the fundamentals that set the stage for this tiny, epic battle. Now, this first rule is so important to get right. It's totally possible to have an infection, where a microbe is just hanging out and growing in your body without actually having an infectious disease. The disease part only kicks in when that microbe or the toxic stuff it makes actually starts messing with your body's normal functions. That's when you get a change from your normal state of health. So, how do we even measure a pathogen's threat level? Well, we use two key terms. Pathogenicity is basically a yes or no question. Does this thing have the ability to cause disease? But virulence, that's where it gets interesting. That measures the degree of harm. Think of it like this. Pathogenicity is whether something is a weapon or not. Virulence is whether that weapon is a water pistol or a fire hose. And this chart, this perfectly shows the timeline of what happens when we do get sick. First, you've got the silent incubation period. The pathogen is multiplying, but you feel totally fine. Then comes the prodromal stage. That's when you get those vague, mild symptoms where you just feel a little off. That ramps up into the illness period, the peak of the battle, where symptoms are at their absolute worst. And then finally, hopefully, you enter convalescence, the recovery stage. And during that illness period, we talk about signs and symptoms. Here's the difference, and it's simple. Signs are the objective things a doctor can actually see or measure, like a fever on a thermometer or a rash on your skin. Symptoms are what you, the patient, feel. They're subjective, like pain or nausea. A doctor is looking for a specific collection of both signs and symptoms, and that collection is what we call a syndrome. Okay, so we've got the rules down. Now for the first play in the playbook, transmission. This is all about how a pathogen makes the journey from wherever it lives to find a brand new host. And why does it want a host? Well, a host provides everything it needs: nutrients, energy, protection, and a way to spread to even more hosts. So every pathogen has what's called a reservoir. It's like its home base. This can be animate, meaning alive, like other people or animals, or it can be inanimate, like soil or water. And to get from that reservoir to you, it often uses a vector. Think of it as a microbial delivery service, like a mosquito or a tick. The really wild thing is that a good vector isn't harmed by the pathogen it carries. It's just along for the ride. So, what are the highways these pathogens are traveling on? Well, there are 5 main routes. Through the air, by direct or indirect contact, through a contaminated vehicle, like food or water, carried by a vector, or passed vertically from a mother to her child. And here we can see 2 of those routes in action. On the left, a classic example of vector-borne transmission. The mosquito, a notorious spreader of some really virulent diseases. And on the right, we've got vertical transmission. This is where an infection is passed directly from a mother to her child during pregnancy, which results in what's called a congenital infection. Okay, so the pathogen has made the journey, it's arrived at the host, but just showing up isn't enough. The next absolutely critical play is to get inside and, most importantly, to stick. This is where a really cool concept called tropism comes in. See, a pathogen can't just attach itself anywhere it wants. It has specific molecules on its surface that act like tiny keys, and they have to find the exact right locks, which are receptors on your cells. That's why a respiratory virus has keys for your lung cells, not your skin cells. It's incredibly specific. But getting to the right cell isn't the only problem it has to solve. It's also a numbers game. You have to ask, how many microbes does it actually take to make you sick? That's what this number is all about. This concept is called the infectious dose 50, or ID50. And this graph explains it perfectly. It's the exact number of microbes you need to expose to a group of hosts to make 50% of them sick. So for strain A here, it only takes about 3,000 organisms. This is why you can be in a room with a sick person and not get ill. You just might not have inhaled a big enough dose. And here's the takeaway. The lower the ID50, the more efficient and infectious that pathogen is. So how do they stick? Oh, they have an amazing arsenal of tools. Bacteria might use these little hair-like things called fimbria or sticky capsules, while viruses use protein spikes, but they all do the same thing. They act like a kind of molecular Velcro, making sure that once the pathogen makes contact, it doesn't just get washed away. Okay, once it's attached, the pathogen faces its biggest challenge yet. Your body is a brutally hostile environment. Your immune system is basically on patrol 24/7, designed to seek and destroy invaders. So to succeed, a pathogen has to be a master of evasion. And their tactics are just so clever. Some pathogens are intracellular, which means they literally hide inside our own cells. Others build these protective capsules so they can't be eaten by our immune cells. They can burrow under mucus, squeeze between our cells, even secrete toxins to kill our defenders. And then there's one of their most sophisticated strategies, building fortified communities. This right here is one of the most brilliant survival strategies in the playbook. Bacteria can actually team up and secrete this slimy, protective matrix, and that forms what we call a biofilm. This fortress makes it incredibly difficult for antibiotics and immune cells to get to them. I mean, bacteria inside a biofilm are exponentially tougher to kill than ones that are just free-floating. All right, let's recap. The pathogen has arrived, it's attached, and it has survived the first brutal attack. Now, for the endgame, causing the actual damage that we experience as disease. So, pathogens mostly cause damage by producing toxins, and they come in two main flavors. Exotoxins are proteins that are actively pumped out by living bacteria. They can travel all through the body and are often incredibly potent. Endotoxins, on the other hand, are actually part of the bacteria's structure, and they only get released when the bacterium dies and breaks apart. Now, some of those exotoxins are particularly nasty. They're called superantigens, and they completely break the rules of engagement. Instead of a normal, targeted immune response, they trick the body into launching a massive, system-wide panic attack. This cytokine storm floods your body with so many alarm signals that your own immune system starts causing all the damage. It's a terrifying case of friendly fire. And this brings us to the ultimate danger of endotoxins, septic shock. When a huge number of bacteria die and release all their endotoxin at once, what we call sepsitemia, it triggers a catastrophic chain reaction. The immune system just goes into absolute overdrive, leading to widespread inflammation, a huge drop in blood pressure, and a domino effect of organ failure. It is one of the most dangerous emergencies in all of medicine. So there you have it. We've walked through the entire playbook: transmission, attachment, evasion, and damage. And knowing each of these steps, every single link in that chain, well, that gives us power. It lets us ask the most important question of all. Where is the weakest link and how can we break it? That's the pathogen's playbook, explained.